Okay, fellas. So this is part two of the video for section 16.1 that DNA is the hereditary material. Okay. So let's talk about the molecular structure of DNA here. We know that DNA is a polymer. So it's made up of many, many repeating units. And each of those units is known as a nucleotide. Each of the nucleotides consists of three parts: a nitrogenous base, a sugar, which is we now know is deoxyribose, and a phosphate group. Now, the nitrogenous bases come in four different flavors: adenine, thymine, guanine, and cytosine. Now, in the early 1950s, a guy by the name of Joe Chargaff, I should say, reported that DNA composition is different from one species to the next. So the DNA composition of a human is different from that of a puppy dog, which is different from that of a parakeet, which is different from that of a fruit fly, and so on. And that gave even more credibility to the idea that DNA was the material responsible for inheritance. Now, Chargaff founded two rules that are now commonly accepted. First, that the base composition of DNA between species varies significantly. And secondly, in any species, regardless of what you're looking at, the number of adenine and thymine bases is equal, and the number of guanine and cytosine bases is equal. Okay. Now, they didn't understand why this was, and Chargaff couldn't explain it, but he did know that if you had 100 A's, you also had 100 T's. And if you had 200 G's, you also had 200 C's. Come on. There we go. So, what does a DNA molecule actually look like? Well, this shows all of the atoms. And we're going to start down here at what we call the 3' end. Now, it's called the 3' end because the links go from the fifth carbon. Notice the numbers of the carbons: 1, 2, 3, 4, 5. Okay, through the phosphate to the third carbon. Notice that's the third carbon right there, right? So this is called the 3' end, whereas this up here is called the 5' end of the molecule. Now we go. So starting at the 3', we go sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. But to the 1' carbon here is attached a nitrogenous base, be it adenine, guanine, or cytosine, guanine, and thymine. Notice the structure here. Thymine and cytosine are both composed of one single ring, whereas guanine and adenine are composed of two rings each. All right, this is going to be important later on. Again, go back to the original PowerPoint that's uploaded into the portal and watch this animation. I don't want to take time to do it here, but it's a great little video to watch and to see. Now, once everyone had accepted the idea that DNA was the material of inheritance, we had to figure out how its structure works and how the structure of DNA actually allowed it to pass on information from one generation to the next. And Wilkins and Franklin is pictured here, used X-ray crystallography to study molecular structure. And this was the picture that they got of a DNA molecule. Now, I have to admit to you, I don't know anything about X-ray crystallography, and I have no idea how that thing gives you the impression that DNA is a double helix, but apparently people smarter than I figured this out, and so that's where the image comes from, and Franklin is responsible for figuring out the structure of DNA, the double helix. And so Franklin's images allowed Watson and Crick to deduce that DNA was helical. And they also allowed Watson to see just how wide the helix was and the spacing of the nitrogenous bases. Go back to what I said a minute ago and think about one ring versus two rings. Okay, the spacing is important. And so there we go. Now, it turns out that thymine and adenine, and we didn't know this at the time, but thymine and adenine both form two hydrogen bonds. So they're attracted to one another. They're not physically bonded, but they're held together by hydrogen bonds. Guanine and cytosine each form 3 hydrogen bonds. In addition, count up the number of rings. I got 1, 2, 3 for adenine and thymine. 1, 2, 3 for guanine and cytosine, and the same for down here. So thymine and adenine can only go together because their spacing is 3 rings and there's 2 hydrogen bonds. Guanine and cytosine can only go together because there's 3 hydrogen bonds and 3 rings. So that's why this all works. The other thing I want you to notice, about this picture is that although the 5' end is on top on this side, it's the 3' end on the other side, and so the double helix is inverted. Now, this is the way that you'll often see these DNA molecules shown. In simplified images. Rather than showing all of the phosphates and sugars, a lot of times the ribbons that you see here will represent the sugar phosphate backbone, okay? And oftentimes they'll be shown in this form. But the nitrogenous bases often come into question, or we want to talk about them, and so oftentimes we'll keep these or them in our pictures, all right? Sometimes, however, even that isn't that important, and we eliminate them, and sometimes we just draw them as 2 ribbons, okay? Again, have a look at this animation in your in the PowerPoint that I uploaded to the portal. It does an excellent job in showing just how long just how the structure of DNA actually works. Okay, so Watson and Crick then built their model, this double helix that was supported by Franklin and her work, and so Franklin had concluded that 2 outer sugar phosphate backbones with the nitrogenous bases in the middle. And Watson's and Crick's model actually showed that. But they also showed, just what I pointed out in the previous slide, that they are anti-parallel, that their subunits run in opposite directions. If you're following one from 5' to 3', the other one runs from 3' to 5'. At first, Watson and Crick thought that the bases were paired alike, A with A, T with T, and so on. But it didn't work that way. Instead, because of what I said earlier about the hydrogen bonds, if you pair a purine with a pyrimidine, you always get a 3-ring rung on the ladder. All right, a 3-ring rung on the ladder, and that's consistent with what Franklin discovered in her images. The reason for that is that if you put 2 purines together, you get a 4-ring rung, and that's too big. If you put a pyrimidine with another pyrimidine, you get a 2-ring rung, and that's too small. And on the other hand, if you have a purine and a pyrimidine together, you get a 3-rung ring. And like Baby Bear said, that's just right, okay? And it's also, of course, consistent with the X-ray crystallography data. All right, so Watson and Crick realized that the pairing was actually more specific, dictated by the structure of the bases, and realized that the reasons that you always got Chargaff's rule of equal amounts of adenine and thymine was because adenine and thymine were paired with one another. And the reason that you got equal amounts of cytosine and guanine was because that cytosine and guanine always paired with one another. And that's shown, we now know today, that's because of the hydrogen bonds that you see here between the molecules. But that's why it works. Okay. So I went through that fairly quickly, but go back and please watch the animations in the PowerPoint. And in addition to that, have a great day. I will see you tomorrow.